Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's dive in. Imagine trying to find something completely invisible, hiding right there in a room. That's kind of the challenge with curing HIV, isn't it? We have amazing treatments, these antiretroviral therapies.
Absolutely life-changing drugs.
Right, they keep the virus suppressed, people live long lives, but the virus doesn't actually go away.
No, it hides. It creates this reservoir.
How does it do that? Where does it hide?
It tucks copies of itself, its genetic material, inside certain white blood cells. Specifically, these immune cells called CD4 positive T cells.
And these are the cells the virus usually infects anyway. Right?
Exactly. But some of these infected cells become resting or, quiet. They're not actively making virus.
So the drugs can't see them, or the immune system.
Pretty much. The virus's genetic code is integrated into the cell's own DNA, just sitting there silently. Current drugs mostly target the active virus replication cycle.
So the virus is inactive.
The drugs don't work on it, and the immune system often doesn't recognize the cell as infected.
And that's the core problem for the nearly 40 million people living with HIV. It's why therapy is lifelong.
Yes, because if treatment stops, that hidden virus can wake up and start making new copies. It's a huge barrier. And tragically, UNAIDS data from 2023 showed we were still losing someone every minute to AIDS-related illnesses.
So that brings us to the topic today. There's been some exciting news, a potential breakthrough maybe?
Well, yes, potentially a big step.
Yeah.
It's about a new way to try and force that hidden virus out of hiding, make it visible again.
This comes from a team in Melbourne, Australia.
That's right. Researchers at the Peter Doherty Institute for Infection and Immunity. We're looking at reports in health news, but also digging into the actual scientific paper they published. It's in Nature Communications.
Okay, so our mission here is to really unpack what they found. Why are researchers saying they felt overwhelmed by these results? What's the buzz about?
It really centers on finding a way to reach that hidden virus, the latent reservoir, particularly in those resting CD4 T cells.
So before this, how did scientists try to tackle this? You hear about shock and kill, right?
Yes, that's been the main strategy for a while. The idea is you use something, a drug, a molecule, to shock the latent virus awake.
Force it to start making noise, basically.
Exactly. Force it to start transcribing its genes, making RNA, making proteins, so the cell looks infected again.
And then the kill part.
Then, hopefully, the immune system can recognize and kill the newly revealed infected cell, or maybe another therapy could target it.
But those earlier attempts, they didn't quite get us there.
No, they haven't cleared the reservoir. The paper we're discussing suggests some reasons why. Maybe those older agents, latency reversing agents they're called, weren't potent enough.
Not a strong enough shock.
Or maybe they only started the process, like initiating transcription, but didn't push the virus through all the steps needed to make it fully visible. Elongation, completion, splicing the RNA properly.
Okay.
And a big issue was specificity. Many early agents weren't very targeted. They'd activate lots of T cells, not just the infected ones.
Which could cause side effects.
Right. Inflammation, toxicity, potentially harming the immune system you actually need for the kill step.
Okay, so that sets the stage for this new research. And it involves technology many of us are now familiar with thanks to COVID.
Exactly. Messenger RNA or mRNA, and those tiny fat bubbles that deliver it lipid nanoparticles, LNPs.
The delivery trucks for the mRNA instructions.
Precisely. The lipid nanoparticles protect the fragile mRNA and help get it inside cells, where the cell's machinery can read the instructions and make a protein.
But the Melbourne team didn't just use any old LNP, did they?
No, and this is key. They specifically designed and developed a new formulation. They call it LNP X in the paper.
LNP X. What's special about its recipe?
They detail the composition, mentioning specific lipids like one called SM-102, which is an ionizable lipid, and also a cholesterol analog called beta-sitosterol. The exact formulation seems critical. They didn't just grab something off the shelf.
So they engineered a custom delivery vehicle. Why was this LNP X formulation such a breakthrough for the HIV problem?
Okay, this is the core finding. The paper reports that LNP X showed, well, they call it unprecedented potency. It was incredibly effective at delivering messenger RNA into those primary resting CD4 positive T cells.
The very cells where the virus hides, and which are normally hard to get things into.
Exactly. Traditionally, getting materials like mRNA into these resting cells is really inefficient. You often had to activate the cells first, which you don't want to do broadly.
But LNP X manages it without that.
Yes. High-efficiency delivery into resting cells without needing prior activation and, crucially, without causing general toxicity or widespread immune activation.
Yeah.
Huge.
It's like a stealth delivery system for these quiet cells. How does it work so well? Did they figure out the mechanism?
They looked into it. The paper suggests it's down to two things. First, increased association. The LNP X particles seem to bind better to the resting T cells and get taken up more efficiently.
Okay, better docking and entry. What's the second thing?
Increased efficiency of translation. Once the messenger RNA gets delivered inside the cell into the cytoplasm.
Translation. You mean the cell actually using the mRNA instructions to make protein?
Exactly that. Transcription is making the mRNA copy from DNA. Translation is the ribosome reading the mRNA to build the protein. LNP X seems to boost how well that happens.
How?
The thinking is it might help the mRNA avoid getting trapped inside cellular compartments like endosomes, and once it's free in the cytoplasm, the cell's machinery is just better at reading it and churning out the protein. So it's not just delivery into the cell, but efficient use of the message once it's there.
Wow. Okay, so they had this amazing delivery tool, LNP X. What message did they decide to send into these hidden reservoirs? What mRNA did they package inside?
They tried two main approaches, two different mRNA payloads designed to act as latency-reversing agents.
To wake the virus up.
Right. The first one was mRNA for the HIV TAT protein.
TAT. What's that?
TAT stands for transactivator of transcription. It's a protein the HIV virus itself makes naturally. Its job is to massively ramp up the virus's own gene expression. It's like a turbocharger for making more virus.
Clever. So using the virus's own tool against it.
Precisely. They packaged the instructions for making TAT protein into LNP X and delivered it ex vivo, meaning in lab dishes, using cells collected from people living with HIV whose virus was suppressed by therapy.
And what happened in the dish?
The results were pretty dramatic. This TAT-LNP-X combination potently induced all stages of HIV transcription initiation, elongation, completion, splicing. They saw lots of correctly processed viral RNA.
A clear sign the virus was waking up and getting ready to produce particles.
Exactly, and this effect was significantly stronger than what they saw using standard lab methods to activate T cells, like mitogens. Plus, a really important point, unlike the general activators, the TAT-LNP-X didn't cause widespread, non-specific T cell activation. It seemed more targeted.
Okay, that's one approach. What was the second message they said using LNP-X? You mentioned CRISPR.
Yes, they use LNP-X to deliver messenger RNA encoding the components for a CRISPR activation system. Not the gene editing kind that cuts DNA, but CRISPR.
CRISPR activation, so
Yeah.
turning genes on.
Right. This system uses a modified Cas9 protein here, dCas9VP64, linked to an activator domain and specific guide RNAs.
And the guide RNAs tell it where to go.
Exactly. They designed the guide RNAs to target the HIV promoter region. That's like the virus's main on switch buried in the human DNA. The CRISPR machinery gets delivered there by LNP-X and basically forces that switch on.
So a very direct way to target the virus's control panel. Did that work too?
It did. It also successfully activated HIV transcription ex vivo. This showed LNP-X could deliver more complex payloads, multiple mRNAs needed for systems like CRISPR.
How did its potency compare to the TAT approach?
In this study, the TAT mRNA seemed more potent at activating the virus, but the CRISPR approach is potentially even more specific to HIV, targeting the integrated virus directly. And importantly, like the TAT approach, it didn't cause that unwanted general cellular activation.
And all this explains why the researchers felt, overwhelmed getting mRNA into these specific cells so effectively.
Yeah, it really was a major hurdle. People had been trying for years. The paper states they achieved over 75% efficiency in delivering mRNA into these resting T cells with LNP-X. That level of success was, as they said, previously considered almost impossible, maybe just a dream.
So stepping back from HIV for a moment, this LNP-X particle seems like a powerful tool just for working with T cells in general, right?
Absolutely. The implications could be much broader. The paper points this out. LNP-X could become a standard tool for lab work, for in vitro delivery to T cells.
For developing other therapies.
Yes, potentially any T cell-based therapy or immunotherapy. It might replace older, clunkier methods like nucleofection or using viruses to deliver genetic material in the lab. It could speed up research. Dr. Roche, one of the researchers, was quoted suggesting potential for other diseases involving white blood cells, even some cancer.
Okay, huge potential there. But let's bring it back to HIV and the shock and kill goal. They achieved a powerful shock, waking the virus. What about the kill? Did waking the virus lead to the cells dying off in their experiments?
Well, this is a really critical point and a limitation they openly discuss in the paper.
Okay.
While the TAT LNPX was great at activating transcription, even leading to some virus particle production ex vivo, they found it did not significantly reduce the amount of intact proviral DNA over the course of their study.
The proviral DNA, that's the blueprint integrated in the cell's DNA.
Yes.
Yeah.
So waking the virus up wasn't enough on its own to get rid of that blueprint or kill the cell in their lab setup.
Why not? If the virus is active, shouldn't the cell die or be targeted?
It's complex. The authors suggest these latently infected cells might actually be resistant to dying. They might have internal mechanisms, maybe overexpressing certain survival proteins that protect them even when the virus reactivates.
So the shock worked, but the kill didn't automatically follow.
Not in this ex vivo system, no. And this is echoed by outside experts, too.
Like Dr. Jonathan Stoya mentioned in the news report.
Right. He wasn't involved in the study, but commented that forcing the virus out is a major step. But the big unknown remains, how do you best kill those cells once they're revealed? Do you need to get every single one? That's still a huge question.
And the study authors agree.
Completely. They stress the need to combine this LNP X-mediated latency reversal with other strategies, things that specifically target the kill step.
Like what?
Maybe drugs that make the infected cells more sensitive to dying, or ways to boost the patient's own immune response, like specialized T cells or antibodies to clear those reactivated cells.
So what are the essential next steps for this LNP X technology then?
Well, the big one is moving to in vivo studies, testing in living systems, starting with animal models.
To check safety.
Safety is paramount, absolutely. But also to understand how LNPX behaves in a whole organism. Where does it go in the body that's biodistribution? How long does it last its half-life? This is all crucial for figuring out doses and delivery for potential human use way down the line.
And they might need to target it better in vivo.
They suggest that, yeah, maybe that's another possibility. Delivering mRNA instructions to, say, disable the CCR5 receptor that HIV uses to get into cells in the first place. LNPX opens doors for delivering various nucleic acid therapies to T cells.
Okay, so let's try to wrap this up. The core achievement here is what?
It's the development of this novel lipid nanoparticle, LNPX. It solves a major technical challenge, efficiently delivering messenger RNA into those key hiding spots for HIV, the resting CD4 T cells.
Which is a huge step forward for the shock part of shock and
It's
kill.
a really significant step, yes. It overcomes a barrier that was proving incredibly difficult.
But, and this is important, it's not a cure yet.
Definitely not a cure yet, but it's a very exciting advance. It gives researchers a powerful new tool and opens up new strategies for combination therapies, pairing this potent way to wake the virus with effective ways to eliminate the infected cells. It renews hope for finding ways to clear that latent reservoir.
It's fascinating. They've cracked a really tough delivery problem for T cells. Which makes you think, beyond HIV, which is obviously critical, where else could this technology make a difference? Being able to get specific instructions into these vital immune cells. What other diseases might benefit down the road?
That's the big question, isn't it? The potential reach could be quite broad, something definitely to keep an eye on.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.